Paul.
And I'm Small.
And today we're doing Khan Academy's MCAT Prep Cell section, cell-to-cell interactions questions. First question. Extracellular fluids are least likely to move through the space between cells that are joined by which type of intracellular junction? A tight junction, a desmosome, macula adherens, or gap junction? I think it's tight junctions. It might just be because the word says tight, but I also kind of don't remember desmosomes or macula adherens.
Yeah, I want to read the answers to these because I don't remember those as well. But
Okay.
I remember the tight junction cells are the ones that allow us to keep that blood-brain barrier. And so they're really important. So if they're asking you a concept question that's like, how do we separate these two things? And it's talking about cell-to-cell barriers, it would definitely be tight junction.
Okay, and tight junction was the correct answer, but like you said, let's look through those hints because we're not entirely sure. Okay, so gap junctions act like tunnels or pipes between cells. Isn't gap junctions how, I think it's cardiac muscle, like pushes nutrients through, like between cells?
I remember something about that, possibly, yes.
It's some kind of muscular cell that has like openings.
Yeah.
For maybe it's like nitric oxide to move through. I don't remember. Somebody tweeted us at the Twitter we don't have the answer to that. Okay, so gap junctions act like tunnels or pipes between cells. Two cells joined by a gap junction are not packed tightly enough to prevent the flow of extracellular fluids between the two cells. Macula adherens and desmosome are actually two names for the same thing. A protein connection between cells that is not tight enough to prevent the flow of extracellular fluids. Okay, so the definition is just that it's not tight enough to prevent the flow of extracellular fluids. So also, these two answers both mean the same thing. Okay, zonula occludens is another name for a tight junction. This type of intracellular junction prevents the passage of fluids between cells. Okay, so it looks like macula adherens and desmosomes, they are just junctions that do allow for the passage of fluids, which could also be in like a passage type thing. If they're asking you, based on this tissue sample that we see fluids flowing through it, we see solutes or something flowing through it, what type of junction is most likely there? You would pick a desmosome or macula adherens. All right, cool. Question number 2. In cardiac muscle tissue, what is the primary purpose of intercalated discs? Oh, this is the thing.
Do you know it?
I think I do.
Okay.
I think this is the thing I was just describing. Permitting the passage of ions between cells, preventing separation of muscle cells during contraction, generating electrical impulses in cells, depolarization and repolarization of cells. So I think this permits the passage of ions between cells.
And you were right.
Okay, very cool. What do we got in these hints? So they must be able to coordinate contraction, which is why the ions need to flow through them. For contraction to be coordinated, an action potential must be able to rapidly spread from cell to cell. In cardiac muscle cells, intercalated discs permit the passage of ions between cells, allowing for an action potential to spread very rapidly to produce a coordinated contraction.
Okay, intercalated discs.
Yeah,
So
I don't really know what that means.
Yeah, I just remember like intercalation just makes me think of ethidium bromide and what it does to DNA.
Oh, that's right. It intercalates. Oh, yeah.
So these junctions, these discs, they just go between cells and they just are just like a channel?
Yeah, if you think about cardiac muscle, like think about the cells like they're two tubes.
Yeah.
Where the two tubes would butt up against each other, there are intercalated discs that act as kind of like funnels between them, or just little holes that permit the passage of ions between the two tubes.
Okay. So I know that cardiac is striated. Is that the striation that we see, those discs?
Yeah, I am almost 100% sure that it
Okay.
is in fact those.
So why do we not have them in muscle cells that are also striated?
Skeletal muscle?
Yeah.
I thought we did, but
Okay.
I don't know.
Okay.
That is why musculoskeletal physiology is something that we all need to go back and review, which we're actually doing a physiology review soon, and I think I picked musculoskeletal
I'm excited.
because I didn't know anything about it. That's
Yep.
going to be a really fun day.
It is. It might have to be like a weekend.
Oh yeah. And that's also, for anyone listening to this part of this podcast, that's a really easy thing to do with a small group of people, is just split up. Like, there's probably like 20 major areas of physiology you're going to need to know for the MCAT. Just split it up across a couple friends. Everyone just become an expert in something you don't know, and then teach it to everybody else.
Yep, that's a really good suggestion. And also, we've listened to 3 or 4 different sources that say keep your group really small. Keep it to like no more than 4 people. That way nobody gets distracted. Everyone is held accountable.
You don't want social loafing to occur.
Oh, no.
Psych. So...
Oh.
Okay, next question. What is the correct order of steps in a common G protein coupled receptor signal transduction? The answer does not include all steps.
Okay.
So the steps that they give us are formation of ligand receptor complex, release of neurotransmitter into synapse, alpha subunit regulates target protein, intracellular phosphorylation cascade, G protein acquires GTP, dissociation of G protein subunits, cross phosphorylation of G proteins, hormone binds to GPCR. So let's just read through what we think it is.
Yeah. So we can also do that. I may have cheated a little bit. As you were reading that out, I went through, and there's only 4 answer choices, and it says, go in order.
Right.
And so, 2 of them you can rule out almost immediately. It starts from formation of the ligand receptor complex before we've even released a neurotransmitter.
Neurotransmitter?
In the synapse.
Well, don't GPCRs only bind to proteins?
They do, right, but there has to be some sort of like signal in order to send out that ligand, right?
There has to be a signal to send out the ligand.
That's what I was thinking.
So I thought it was hormone binds to GPCR. Because I didn't think that.
Are we not starting from the noggin? Are we not starting from like send out,
I might not be thinking with my noggin.
No, I think you're right. I think I like automatically assumed we would be starting from some like neuronal impulse to send out this hormone. And maybe that's not right. So
you would have chosen the one that starts with release of neurotransmitters. I would have chosen the one that started with hormone binds to GPCR, because there are answer choices that involve both of those. Yeah. So let's read through them. So let's read through yours first.
Oh no, let's not.
Okay, let's read through mine first. So mine would have been hormone binds to GPCR, and then formation of a ligand receptor complex. I'm still okay with that. 5. So G protein acquires GTP. Okay, that still fits. Cross phosphorylation of G proteins. I think that's backwards.
Okay.
Right?
What did it come after? It came after which one?
It said G protein acquires GTP, then cross phosphorylation of G proteins. Well, let's just keep reading to see if it. Okay, so alpha subunit regulates target protein. Yes, that would happen. And then intracellular phosphorylation cascade. Yes, that would happen. So we know it's 3-4 at the end.
Yeah, for sure.
Literally, the only difference between your choice and my choice is which one do you start with. Do you start with a hormone, or do you start with release of neurotransmitters into the synapse? And then the other answer choices both start with the formation of a ligand receptor complex, and then go to G protein acquiring GTP, and then dissociation. Yeah, it doesn't seem like those are correct.
So if I'm thinking about, let me think of an actual example. If I eat a lot of sugar, then my pancreas is going to be like, oh, send out some insulin. I need to bring this into the cells. And that doesn't necessarily get integrated in my brain. So maybe I don't need a neurotransmitter here.
I'm actually maybe going a completely different route here.
Okay.
So I just saw one of the answer choices that says formation of a ligand receptor complex, which could be true if you're just not saying what specific ligand it is. So formation of a ligand receptor complex, then the G protein acquires GTP, then the alpha subunit regulates the target protein, which would be like adenylate cyclase, Then you have intracellular phosphorylation cascade. Then you have dissociation of G protein subunits.
The dissociation of the G protein subunits would happen when that cap comes off earlier, right?
You are so right.
Okay.
I was thinking dissociation of GTP.
Yeah, because it has GTPase activity.
Right.
Yeah.
Okay, so I might go back with my original answer. Oh, which is wrong. Oh, we were both wrong.
Oh, no.
This is unbelievable.
It's the last one.
The correct answer is formation of the ligand receptor complex. Then G protein acquires GTP. Then dissociation of G protein subunits. Then alpha subunit regulates target protein. Then intracellular phosphorylation cascade. Wow, that's right. Why didn't I think... Why was that like the last one on my mind?
So apparently, GPCRs don't only serve to do these specific functions of like neurotransmitter work or hormone signaling, which I thought they were totally only involved with.
What else are they involved with?
I don't know. Other cell signaling, it says reception, transduction, and response. So
not necessarily just hormones or neurotransmitters.
Yeah, but I don't know an example of anything else.
Well, I guess it was safe to say that it's the formation of a ligand receptor complex instead of getting specific and saying hormones or neurotransmitters, because we weren't sure.
We fell for both the traps.
Yeah, we did. Okay, but this answer makes sense. Next question.
Take cell bio.
Take cell bio. Which type of membrane receptor protein best describes receptor tyrosine kinases? Is it a GPCR, an intracellular receptor, an enzyme-linked receptor, or a ligand-gated ion channel? So it's not a GPCR. Mm-hmm. If I remember receptor tyrosine kinase correctly, it's like two proteins. It's a dimer that upon binding of its ligand on the extracellular side, the intracellular side like changes conformation, and you have a bunch of tyrosines that get phosphorylated.
I think so, yeah. And they like dimerize and...
Yeah.
Yeah.
So... is it an intracellular receptor? That seems too specific, and it's a receptor on the extracellular side, technically.
Yeah.
A ligand-gated ion channel?
Mm-mm.
No, it might turn on one, but it is not one. Enzyme-linked receptor. I think there is some kind of enzyme that's always linked to it.
Well, it is a kinase.
That's right. Oh my gosh, you're right. Yeah, and
nice.
That's the correct answer. Okay. Nice process of elimination and thinking our way through that answer.
Oh, and their explanation was exactly what you said, Tal. Well done.
Oh, what is that?
Upon activation, receptor tyrosine kinases dimerize and phosphorylate tyrosine residues on each other's tails.
I feel good about myself.
Yeah. Isn't there an insulin one that does it all in like one?
An insulin receptor tyrosine kinase?
No, there's like a 2-part one, and then there's one that like does it all itself. I'm gonna have to review these.
Yeah, I don't know.
And it might be an example of one of these. I need to review it.
But I think that's another good point, that if you get to the point where you're starting to receptor tyrosine kinases and GPCRs, it's in like if you have time, it would behoove you to learn about like the major ones like insulin.
Yeah.
Or different hormones. Like, is there anything specific that you might want to know about them that could help you in a passage? Because you could probably answer the passage regardless, but it would be nice if you understood the content going in.
Yeah, Tal is really good about going through these passages, and sometimes he doesn't even need to read them. He just knows the background information for that concept, and so then he can answer them like they're discrete questions almost.
Yeah, it really feels like about half the test could be answered like a discrete question. And then a lot of the test you can just actually go back to specific points in the passage that don't require any other context. This is a little bit dangerous, but if you practice enough passages, this is exactly why everyone tells you, like, do so many more passages, do so many more practice tests, half lengths, just do that over content. Like, get your basics on content. And I almost really wanted to go into this entire MCAT just studying the quick sheets. As dangerous as that would be, I want to go in just studying Kaplan quick sheets and then take the test. But unfortunately, I'm going through all of the content and probably going to be way better off for it. Okay, next question. Which signaling molecule is least likely to serve as a ligand for an intracellular receptor? Intracellular receptor. Is it growth hormone, nitric oxide, ethylene, estrogen? I think we're looking for which one of these things is not like the others.
Yeah.
So first off, we have estrogen and growth hormone, which we know those are big.
Yep.
We know that ethylene and nitric oxide are small.
But ethylene, if I remember, is the C2H4.
Yes. So C2H4 would pretty readily cross the membrane.
Yep.
Nitric oxide,
Polar.
Super polar. And then, now that we think about it, aren't growth hormone and estrogen both steroid derivatives?
I don't know about growth hormone. Estrogen, smacks of steroid derivative.
Right.
It looks just like it.
I think growth hormone is too. Not entirely sure, but I know that nitric oxide probably is not going to cross the membrane. Now, if I'm wrong on that, then I think the reason is because it's small. And that just because it's small, it might be able to diffuse, but I'm still going to go with it.
So what we're looking at here is small and polar. Those are the two things we should consider when we're talking about crossing a membrane.
Yeah, it's really the size and the polarity.
So having an idea about that chart that shows you, these things pass very quickly, and then, you go down and you get more polar and larger molecules, and then at the very bottom there's ions that absolutely have to have channels in order to cross.
Yes. So let's check it. Ooh, it wasn't right. I guarantee you it's growth hormone. It is.
How big is it? I don't know.
I don't know either. So we have to keep going through these until we find.
NO is nonpolar.
Nitric oxide is nonpolar.
How?
I thought nitric oxide was NO2. Which
Well, it's just NO, but they have a different electronegativity. Maybe it's not strong enough to be polar.
Oh, maybe. All right, it says growth hormone is a large peptide hormone. Oh,
it's a giant protein. That makes sense.
Well, yeah.
Okay.
So, growth hormone. What do we know about growth hormone? Is that where's that released from? Is it from the anterior pituitary?
Is it one of our flat peg?
Flat peg. Oh, okay.
Growth
Flat
hormone
peg.
releasing.
Oh, G is gonadotropin.
Yeah.
So growth hormone, I don't know where growth hormone comes from, but you know what? Once again, that's a really good thing that, you might know what peptide hormones and steroid hormones do, but then start looking at, the major examples, and I will say, admittedly, growth hormone is probably one that I should have known going into this. But yeah, okay, growth hormone is a large peptide.
In case anyone was wondering, the thing that I'm teaching, one of the things I'm teaching at our physiology weekend is hormones, because I am terrible at them.
We're going to come back being super experts on all kinds of stuff, including hormones.
Mm-hmm.
Okay, next question. Which statement represents a key difference between G protein coupled receptor and a receptor tyrosine kinase? Is it a receptor tyrosine kinase acts as an enzyme to its intracellular substrate? A single ligand can activate multiple signal transduction pathways through a receptor tyrosine kinase. A receptor tyrosine kinase is more likely to activate gene transcription, or a G protein coupled receptor can be activated by a diverse array of ligands. Okay, I think that last one is wrong.
Mm-hmm.
They can both be activated by a diverse array of ligands. I don't think a receptor tyrosine kinase is any more likely to activate gene transcription than a GPCR.
Yep.
A single ligand can activate multiple signal transduction pathways through... No, it can do the same thing through a GPCR. So a receptor tyrosine kinase acts as an enzyme to its intracellular substrate.
Yep, I would agree.
Yeah, because you pointed out to me, I keep saying the name, but I wasn't even thinking about it. It's a kinase.
Yeah.
Which means that its activity is to phosphorylate something.
Yes. And so I always tell people, when you're taking biochem, and if you can really get the hang of enzymes, then that test, including all of the competitive, non-competitive, uncompetitive, all of that stuff,
Right.
If you can get a hang of it, that test is going to be a breeze for you.
Yeah.
Because the names make intrinsic sense, and so do the curves. You don't even have to memorize anything. You can walk your way through it really with reasoning.
And that's thinking like you would think in the MCAT, kind of.
I sure hope so.
All right, and we were correct. Next question. When acetylcholine binds to its receptor on a neuron, sodium rushes out of the cell. Sodium rushes out of the cell? Interesting. Which type of receptor is this an example of? Muscarinic receptor, nicotinic receptor, voltage-gated ion channel, or ligand-gated ion channel?
I'll leave this to the voltage-gated expert.
Okay, so first off, voltage-gated ion channel means that the actual channel, that sodium channel, has specific transmembrane regions that physically move and then change the conformation of the protein to let it in. There is no binding of acetylcholine in that case. Now, a ligand-gated ion channel might be correct.
That is literally the definition of ligand-gated.
Yeah.
Yeah.
Muscarinic receptor. I remember nicotinic receptor is like it's nicotine and something else, like maybe caffeine. Okay. I don't know. But muscarinic receptor, for some reason in my head, that is screaming that is what they call an acetylcholine-gated ion channel.
Okay. Nicotinic and muscarinic receptors are both targets of acetylcholine. Both are channel proteins, and the question does not give enough information to distinguish if the receptor is one or the other.
Oh, okay. So I would go with ligand-gated ion channel. Because I didn't know enough about muscarinic or nicotinic, I would have just gone straight with ligand-gated ion channel. And that's the correct answer.
Okay, I've never heard of those before.
The muscarinic and nicotinic?
Yeah.
Yeah, they mean something. I don't know what they mean, but they mean something.
That's helpful.
It's important to know at least. Okay, next question. In the following epinephrine pathway, which component is the second messenger? So we have an epinephrine pathway. They want to know what's the secondary messenger. Okay, epinephrine leads to GPCR, leads to G protein, leads to adenylyl cyclase, leads to CAMP, leads to protein kinase A. What's the secondary messenger? Is it adenylyl cyclase, GTP, G protein, or CAMP?
Do you remember?
Oh, it's CAMP.
Oh, yeah.
So secondary messenger basically just means that the first messenger is the G protein. The adenylyl cyclase, you wouldn't really call it a messenger. It's a maker. It makes the next messenger, which is the second one.
I wouldn't say that the first messenger is the G protein, would you? I would say it's the ligand.
Oh, well, I guess. Well, I guess if you're talking about the actual the signal transduction pathway,
yeah,
then in my head I would be implying that or assuming that they consider the beginning of the signal transduction to be when the signal is received. So
which is from the ligand.
Yeah, so like the instant the signal is received, that's when the pathway starts. So what's the first thing in the pathway? It's the G protein.
Okay.
That's how I think about it in my head.
Okay.
Because it's the primary messenger. So we said cyclic AMP, and that is correct. All right, next question. Question number 9. A researcher plans to determine the rate of diffusion of a substance across a plasma membrane by measuring how quickly it moves across a single layer of cells. Which type of cellular junction would best accommodate her experiment? Oh my God, did I not say at the beginning of this that this might be a question?
Yes, wait.
Okay, so the possible answers are desmosomal adhesion, intercalated discs, tight junctions, or gap junctions.
So tight we can take out.
Yes, because we remember that definition. So then we have gap junctions, intercalated disc, desmosomal adhesion.
A single layer of cells does not make me think we would have intercalated discs like in cardiac muscle.
Yes.
Because I feel like that would be a complex thing.
Yeah, it normally means you have think about like a tube of like many series of cells that you pass through.
Yeah. And so if I'm just talking about diffusion across one layer of cells, like even my cells on a plate, my MEFs, I'm just thinking maybe gap junctions, like in the gut.
I'm actually a little confused here because, I mean, I would have said gap junctions or desmosomal adhesion.
Yeah, because the other one told us that they both allow things to pass through.
Right.
But I think the gap junctions are specifically like in our intestines, and when we have a simple layer of epithelial cells where we can take in nutrients that way from the apical side.
That sounds good to me.
Okay.
Gap junctions. 1, 2, 3.
Oh no!
Oh no! Yeah, I bet it's desmosomal adhesion. It's not.
Oh no!
Good heavens! Tight junctions. Did we? Okay, we read the question wrong. Obviously.
Yeah, did we?
Okay, a researcher plans to determine the rate of diffusion of a substance across a plasma membrane. Oh no! Oh, that was a big wave file.
Yeah.
Sorry for your ears, everybody. Oh my God. Okay. Please listen to us now. I hope you didn't tune out right before we just had that revelation. But they wanted to know how quickly does it diffuse across a plasma membrane.
Yep.
Meaning, and we should have seen this when we saw there was like, one of these things is not like the others. That if you're trying to go across the plasma membrane,
you don't want anything between. Oh my goodness.
We don't want anything going in between those cells to mess up the experiment. Dang.
Experimental design keeps slapping me in the face.
It's really such a bigger deal in the MCAT than I thought.
Yeah.
Just being able to like read through read through really simple methods and then understand them.
Make sure you get in a lab somewhere.
Definitely. It'll help. Obviously, it's not helping us, but
oh no,
it might help. Who knows? Okay, next question. Steroid hormones, such as testosterone and estrogen, act on target cells by passing directly through the cell membrane. If steroid hormones can enter any cell, how do they only produce an effect in target cells? Is it that only in target cells can testosterone initiate the proper phosphorylation cascade? Only target cells contain the enzymes to appropriately modify steroid hormones? Only target cells possess the proper intracellular receptor? Or only target cells retain the necessary genes for the steroid to act on? Intracellular receptor. That jumps out at me.
Yep. Do you remember what is specific about steroid hormones?
Like what on them specifically makes the action, the like signaling action?
No. Like what action do they have that most other hormones don't necessarily have?
Let's see. It wouldn't be regulate gene transcription because they both do that. It is?
Yeah. So it's saying, both of them can act on target cells by passing directly through the cell membrane. And which is true, they can get through all cell membranes. So how do they only affect the specific tissues they're supposed to? Like, why do we develop as men and women based on, these testosterone and estrogen, different concentrations in men and women? Mm-hmm. And like, how do they act on the specific sex organs the secondary sex organs that develop? And that is because their receptor on the inside of the nuclear membrane has a DNA binding region. So they do act as transcription factors.
Oh, okay. So steroid hormones can bind. Do they bind to actual receptors on the intracellular part of the cell?
Yeah.
And they also can bind directly to DNA?
They cannot bind to DNA, but their receptor has like a lysine finger, or there's like some asparagine one. Uh-huh. So this is kind of silly. I was doing like biochem research one night, reading really, and found that there's actually a band called like Lysine Fingers or something like that. But the DNA binding region of those steroid receptors in the nucleus have those specific like positively charged areas that allows those receptors, not the steroids, to bind to the negatively charged DNA and affect how it's transcribed. It's incredible.
Huh. Very interesting.
Yeah.
Well, once again, difference between steroids and peptide hormones.
Yes. Growth hormone, peptide. Ugh.
Yep. Strong work, gang.